Good afternoon, everyone. Welcome to our lesson on cholesterol and lipoprotein metabolism. Cholesterol is an important steroid and lipid in animal tissues. As a steroid, it means it contains the steroid nucleus, which you can see in the image in the right. As a lipid, this also means that cholesterol is relatively insoluble in water. Human adults create approximately 1 gram of cholesterol per day, which means that even if you do not take in cholesterol from the food, you make 1 gram of it every day through de novo synthesis. We consume approximately 300 milligrams of cholesterol per day through the diet. Cholesterol in the human body is stored as a cholesterol ester, which means it is composed of cholesterol, and attached to that cholesterol is a fatty acid chain. So cholesterol plus fatty acid. Let's take a closer look at the structure of cholesterol. It's a 27 carbon compound, as you can see in the image in the right, and it has a steroid nucleus, which is composed of 4 fused hydrocarbon rings labeled A to D, with letter D, shaded in blue, composed of a cyclopentane. It has an 8-carbon side chain attached to carbon number 17 of the D ring. So that's number 21, 20, up to 27. A hydroxyl group or an OH group is attached to carbon number 3 of the A ring, and a double bond exists between carbon number 5 and 6 of the B ring. So all of these specifications are found in cholesterol. Let's now talk about how the body synthesizes cholesterol. This is known as de novo cholesterologenesis or cholesterol synthesis. The substrate or the starting substance is acetyl coenzyme A. And from this, you know that there are lots of substances in the body taken from the food that can create acetyl coenzyme A. So, our substrate is abundant. Where does it occur? It occurs virtually in all tissues in the human body. Why? Because cholesterol is an important component of the cell membrane. In the cell membrane, cholesterol maintains the fluidity of the cell membrane, which makes it pliable, and at the same time, it makes it strong. It is important, especially in the liver, in the intestines, in the adrenal cortex, ovaries, testes, and placenta. In the liver, it is used to synthesize bile acids, which we need to digest fat. And in the adrenal cortex, the gonads, and the placenta, it is utilized to create steroid hormones. It is synthesized in the cytosol and endoplasmic reticulum. What is the rate limiting step, or what is the slowest step? It's the conversion of HMG-CoA, hydroxy methyl glutaryl coenzyme A, into mevalonate via reduction. So what's the rate limiting step or the slowest step? The reduction of hydroxy methyl glutaryl coenzyme A. The requirement for this reaction is 2 molecules of reduced NADP. So since this is the slowest step in the reaction, if we can alter the kinetics of this reaction, then we alter also the entire cholesterol synthesis pathway. So let's now take a closer look at the image here in the right. Acetyl coenzyme A fuses with acetoacetyl coenzyme A to form hydroxy methyl glutaryl coenzyme A. This is reduced utilizing the hydrogen carried by reduced NADP to form mevalonate, and that is the rate limiting step, catalyzed by HMG coenzyme A reductase, which is the site of action inhibition by statins or HMG-CoA reductase inhibitors, which we use to treat dyslipidemia. Mevalonate then is oxidized to form isopentenyl pyrophosphate. Isopentenyl pyrophosphate combines with other isopentenyl pyrophosphate together with pyrolysis to form farnesyl pyrophosphate. Farnesyl pyrophosphate again combines with other molecules of farnesyl pyrophosphate to form squalene. So starting at isopentyl pyrophosphate, we have 5 carbon units. So these will combine with other 5 carbon units to elongate and create the ring. So 5 carbons, 10 carbons, 15 carbons, and so on. Squalene is then oxidized to form cholesterol. So in essence, the multiple steps are HMG-CoA formation, the committed step, which is the reduction, phosphorylation, and then condensation of these 5 carbon units, and then lastly, cholesterol formation. So let's reiterate the sequence of steps. Acetyl coenzyme A is produced, usually from glucose and pyruvate. Acetoacetyl-CoA and acetyl-CoA condense to form HMG-CoA. HMG-CoA is reduced to mevalonate. Mevalonate is converted to 5 carbon isoprenoid units, and 2 isoprenyl pyrophosphates or isoprenoid units condense to form geranyl pyrophosphate, which is 10 carbon units long. This reacts with another 5 carbon compound to form farnesyl pyrophosphate, which is 15 carbons long. Squalene is formed from 2 farnesyl pyrophosphate molecules, so we have a total now of 30, and it is cyclized, which means we form ring structures, and this produces lanosterol. Finally, lanosterol is converted to cholesterol. How do we regulate cholesterol biosynthesis? So, cholesterol biosynthesis produces cholesterol, but that product in itself causes inhibition of the pathway. So, there is some form of negative feedback. Cholesterol and its metabolites repress transcription of the rate-limiting enzyme, HMG coenzyme reductase, via activation of SREBP or sterol regulatory element binding protein. So if you already have produced cholesterol, the levels of HMG-CoA reductase will go down, and that will also slow down cholesterol biosynthesis. Again, because you already have cholesterol, form of negative feedback. We also have enzymatic regulation. So we can either phosphorylate the enzyme, which makes it inactive, or dephosphorylate an enzyme using a phosphatase, removing the phosphate group, making it active. Or we can regulate the activity of the rate-limiting enzyme through hormones. So insulin and thyroid hormone will speed up the activity of HMG-CoA reductase, which also means it will create more cholesterol if you have insulin and thyroid hormone. And glucagon and glucocorticoids will decrease the activity of the enzyme, which means when you secrete glucagon and glucocorticoids, which I suppose you already remembered, happens when you don't have something to eat, when you're fasting, then the synthesis of cholesterol will also slow down. The cholesterol ring that we have formed cannot be metabolized in humans. So we cannot degrade the entire cholesterol. So what we do is convert that cholesterol molecule into other products. For example, conversion to bile acids and bile salts, which will be found in the bile, which allows you to solubilize dietary fats. But eventually, these bile acids find their way in the feces where they are secreted. You can also secrete cholesterol into the bile itself, and this is transported into the intestines where bacteria, which has the appropriate enzymes to degrade the cholesterol ring, converts it to coprostanol or cholestanol. So, in summary, we can't degrade cholesterol, so we secrete it. Let's now go to lipoproteins. Remember that cholesterol is a lipid. And you have also been aware that there are lipids found in the blood. And blood is made up of plasma. It is largely made up of water. So you have a hydrophobic substance present in plasma, which is water. So how does that happen? It's through lipoproteins. Lipoproteins are spherical, macromolecular complexes of lipids and proteins called apolipoproteins. So to make it simple, a lipoprotein is made up of lipid and protein. That protein is called an apolipoprotein or apoprotein. So, the core of the lipoprotein is usually a neutral lipid in the form of triglyceride and cholesterol, and the shell of the lipid is usually amphipathic apolipoproteins, which means the proteins. So they are found in the shell or in the periphery of the lipoprotein molecule. We also find in the shell phospholipids, which have a hydrophilic portion, and free fatty acids. Functions of lipoproteins include keeping the lipids soluble in plasma. It also provides an efficient transport mechanism for lipids to and from various tissues. So, what this means is that the lipid, for example, in the liver, can find their way into other portions of the body through a transport mechanism, and that is the lipoprotein. Okay, so let's now focus our attention on the image here in the right. At the top image, it tells us a generalized structure of a lipoprotein. In the core of the lipoprotein, we find the neutral lipids, those that are in brown and yellow. These are triglycerides and cholesterol esters. And at the shell of the lipoprotein or the periphery of the lipoprotein, we find some unesterified cholesterol, again because we have a hydroxyl group in cholesterol and that can be polar. We have phospholipids, which have polar components, and apolipoproteins, which are made up of amino acids, and these are also polar. So, another illustration is found here below, which makes it more simple. In the core, again, would be the triglyceride and cholesterol esters. And you have the peripheral apoprotein, which is in pink. And these apoproteins are special because they're not only structural components, but they also act like enzymes. So you have free cholesterol in the shell, and you have integral apoproteins. So we have peripheral and integral apoproteins. Peripheral apoproteins, they float in the lipoprotein molecule, whereas integral apoproteins insert themselves deeply into the lipoprotein molecule. So let's now talk of important lipoproteins. First would be the chylomicron. It's produced in the intestine and it's 99% lipid. It's the largest lipoprotein and it has the highest triacyl glyceride content. So later, when we talk about the metabolism of chylomicrons, you will appreciate why that is so. We have very low density lipoprotein, and this is produced in the liver. and the lipid component is a little bit lower, 93%. So this very low density lipoprotein circulates throughout the body, and the capillaries of the body continuously remove free fatty acids from VLDL. And so once these free fatty acids are removed, we form intermediate density lipoprotein. And the same thing happens, it circulates, IDL circulates until we form LDL. Okay? So as VLDL circulates, more and more free fatty acids are removed until you form an LDL where a lot of the free fatty acids have been removed, and what is left would be a molecule very rich in cholesterol. That's why it has the highest cholesterol content. And HDL performs reverse cholesterol transport, which means cholesterol found in the periphery is delivered back to the liver. So it decreases cholesterol in the periphery. It has the highest protein content. That's why it's high density, because it has lower lipid content and high protein content. So here in the image in the left, you will see the relative sizes of the different lipoproteins. So at the top, you have the chylomicron, and compare that to the VLDL. And the VLDL circulates, releasing its free fatty acids until it becomes small. That's the LDL, with the central cholesterol core colored orange. And you have HDL, or high density lipoprotein, which is made up of lots of proteins, apolipoproteins, and phospholipids, increasing its density. So remember that apolipoproteins are the protein components of lipoproteins. And again, I told you a while ago that apolipoproteins or apoproteins are not only structural components, but they are also enzymes which catalyze certain reactions or binding to receptors. So your A1, ApoA1, is found in high density lipoprotein, and it functions as a cofactor of lecithin cholesterol acyl transferase, which we will learn later. It's a structural component also of HDL. Apolipoprotein B100 is found in VLDL, very low density lipoprotein, intermediate density lipoprotein, and low density lipoprotein. It mediates the assembly and secretion of VLDL. ApoB48 is found in chylomicrons, and it is the main apoprotein which causes secretion of the chylomicron. ApoC2 is found in all of the lipoproteins, and it is a cofactor of lipoprotein lipase. Again, it's important. ApoC2 is the cofactor of lipoprotein lipase, which continuously cleaves off free fatty acids from the lipoprotein. ApoE is found in almost all lipoproteins, and it mediates the uptake by the liver of chylomicron remnants and intermediate density lipoproteins. So let's now go into how all of this information integrates into lipid transport. Again, lipoproteins are ways by which different organs of the body transfer lipids between each other. So let's now learn the metabolism of chylomicrons first. So let's start with number one. The location is the small intestine. Chylomicrons are produced by intestinal mucosal cells. So what happens first here is that the lipase that is secreted by the pancreas hydrolyzes the dietary lipids, releasing free fatty acids. Those free fatty acids are absorbed by the intestinal epithelial cell, and the intestinal epithelial cell rearranges those free fatty acids to form a chylomicron. And those intestinal epithelial cells then secrete the chylomicron into the lymphatic capillaries. So we have that's step one. You absorb, you package the lipids into chylomicrons. Step two, ApoC2 and ApoE are transferred from HDL to the immature chylomicron. So initially, the chylomicron does not contain ApoC2 and ApoE. So the chylomicron receives the ApoC2 and ApoE from HDL. After receiving those two apoproteins, you now have a mature chylomicron. The chylomicron again is secreted into the lymphatic capillaries, but eventually they find their way into plasma. So in the plasma, that chylomicron encounters the enzyme lipoprotein lipase. So the lipoprotein lipase, by the term lipase, hydrolyzes the triglycerides, releasing free fatty acids which are absorbed by the peripheral cells, for example, fat cells, for example, muscle cells, and they also release glycerol. So by the action of lipoprotein lipase, we have degraded triglycerides into free fatty acids and glycerol. After releasing all of the triglycerides or almost all of the triglycerides, what is left would be a chylomicron remnant. This chylomicron remnant then encounters the ApoE receptor of the liver, and it binds there, and the liver then takes up the chylomicron remnant and processes it, degrading all of the lipids found in the chylomicron remnant. So that is how the chylomicron transfers lipid from the food that you ate into the blood, into the peripheral tissues, and into the liver. Next would be the metabolism of VLDL. So here we start with the liver. The amino acids in the blood and the free fatty acids and the lipids in the blood are repackaged by the liver into very low density lipoprotein. So step one, the liver secretes nascent triacylglycerol or triglyceride rich VLDL particles. So like your chylomicron, the initial VLDL does not contain ApoC2 and ApoE. So the VLDL will have to first receive the ApoC2 and ApoE from HDL to become a mature VLDL. And like the chylomicron remnant, the VLDL traverses the capillaries where it encounters lipoprotein lipase. And like chylomicrons, VLDL, by action of the lipase, the triglycerides are hydrolyzed and you release free fatty acids to the peripheral tissues and glycerol back to the liver. After releasing these lipids, what is left would be intermediate density lipoprotein. So that intermediate density lipoprotein can go directly to the liver where it will be repackaged, or it can continuously circulate through the capillaries until it becomes more dense in the form of low density lipoprotein, from VLDL to IDL to low density lipoprotein. So you have to understand that the VLDL molecule has lots of triglycerides, but as it circulates, the amount of triglycerides that it holds goes down until what is left in the low density lipoprotein is a lipoprotein very rich in cholesterol. The extrahepatic tissues, the peripheral cells, have specific receptors for LDL. So they have receptors for ApoB100, which allows the different tissues of the body to absorb the LDL and release from its core the cholesterol. This cholesterol will then be utilized by the peripheral cells to make the cell membrane or to make hormones. Okay? So that's the metabolism of VLDL, similar to a chylomicron, but what is left is LDL. Next, what happens here is the cellular uptake and degradation of LDL. This is the terminal portion of the illustration a while ago. So LDL with its apolipoprotein B100 binds to the LDL receptor in the cell membrane of the peripheral cell, which recognizes ApoB100. A vesicle is formed, and this vesicle fuses with that peripheral cell's lysosome. And remember that lysosomes contain hydrolases, which are enzymes that perform hydrolysis. So, your LDL molecule is now in the lysosome, it's hydrolyzed, and what is released would be amino acids, fatty acids, and cholesterol. That cholesterol will inhibit de novo cholesterologenesis in that cell, but at the same time, it can form the different functions of cholesterol in the cell as components of the cell membrane or as precursors for certain hormones. Next and last would be the metabolism of HDL, high-density lipoprotein. So we start with the liver secreting high-density lipoprotein. So from the top left of the image, the liver secretes nascent HDL or immature HDL. So this form of HDL is still discoidal, which means it's like an oval structure. So this discoidal nascent HDL then takes up free cholesterol from the peripheral tissues through the action of LCAT, lecithin cholesterol acyl transferase. Okay, so it takes up cholesterol esters from the periphery. So as it takes up cholesterol esters from the periphery, it continues to enlarge. Okay, and that large high-density lipoprotein will then be catalyzing the transfer of ApoC2 and ApoE, as what we have discovered from the previous images, into VLDL to make it more mature. So that transfer of ApoC2 and ApoE is catalyzed by CETP, cholesterol ester transfer protein, cholesterol ester transfer protein. And lastly, that HDL, which is now rich in cholesterol after taking the cholesterol from the periphery, goes back to the liver, where the liver degrades the HDL and utilizes the cholesterol. So by the metabolism of high-density lipoprotein, you now have a way of transferring cholesterol from the periphery back to the liver. So through this lecture, we have learned, albeit through overview, cholesterol metabolism, how the body creates cholesterol, how the body transports lipids, and in particular, cholesterol. So through our application lecture, we will understand how it is important for us to understand the nooks and crannies of lipoprotein metabolism. So thank you for listening.